Here's the simple workflow for a typical CRISPR experiment for mammalian cells. This video is about the guide RNA design using some freely available online tools. I'm going to demonstrate one type of workflow to design CRISPR guides. Once you have a target location in mind, you open up the UCSC genome browser and navigate to that location. Here I have the human genome HG38 assembly pulled up. We are zoomed into the COX9 locus with its acetylation signature. And for this tutorial, say I am interested in targeting somewhere in this tiny area next to the potential enhancer. Let's zoom in. We can get the DNA sequence of this viewable area in the browser by typing VD and then click Get DNA. This is the sequence of the area we zoomed into. So we will design a guide RNA using this DNA as the template. There are many websites, tools, and softwares to design CRISPR guides. I will use CRISPR and CHOPCHOP as examples. We will compare the outputs from both of these tools. In CRISPR, you provide the DNA sequence. Let's give it a name. Choose the appropriate reference genome assembly where you got the DNA sequence from. And for this tutorial, I will use CRISPR Cas9 that targets using the NGG PAM sequence. Then just hit submit and wait for results. CHOPCHOP, on the other hand, takes in the genomic coordinates. Don't forget to specify the assembly for these coordinates. The CRISPR Cas9 used reflects the NGG CRISPR Cas9. And I will just stick to knockout for demonstration. Then click Find Targets. On CRISPR results, you can scroll through the target sequence and check out the distribution of NGGs. The colors of NGGs correspond to the level of their associated guides specificity. If you scroll down, you will find all the hits listed out. There's a lot of information, but let's focus on a few important things. The guide and PAM sequence is the important one. The PAM is separated from the guide with a space. As you can see, there are many metrics in this table. The most important is the specificity score, and you can read about it if you hover over the column name. The higher the number, the better. The guide sequences are arranged such that the highest MIT specificity score is at the top. For each result, there is the associated position and the strandedness of that sequence. When comparing the specificity score, it seems like the top two scores are exactly the same, meaning that there might not be a meaningful difference. If you look at the off-target sites, meaning that if you were to pick a guide where else in the genome it can potentially target, there is a small difference. There are 0 location with 0 mismatches, 0 location with 1 mismatch, 0 locations with 2 mismatch, but if you allow 3 mismatches in the guide and target, you get 4 such spots in the genome. And if you allow 4 mismatches, there are 96 such locations in the genome. And you have this information for each guide. There are more details in the information box, which you can read as well. So, based on all mismatches, there are a varying number of off-target spots for these guides. The high probability off-targets are highlighted in the gray, and you can see that there are very few of those for the top hits. So, based on this non-exhaustive analysis, you can pick any of the top 3 guides. Let's check out CHOPCHOP. You have got the guide distribution on the target DNA sequence, and the top 2 results are highlighted here. For each guide, there is an efficiency metric, which is different from the specificity score. This efficiency is the same as the third column in CRISPR output. Nice thing about CHOPCHOP is the ability to visualize results in the UCSC genome browser. Let's do a quick comparison of top hits. The first result from CRISPR is in the top 3 in CHOPCHOP. The second result from CRISPR is also in the top 3. The third is also in the top 3 in CHOPCHOP. So it seems that both programs are in alignment with the top guide sequences. One small note of comparison. In CRISPR, the PAM and guide are separated with a space, and that is nice to see visually. CHOPCHOP can be a bit confusing because it does not provide a space between the PAM and its guide. Now, you can pause the video and pick a guide that you think is the best. I think any of the top 3 guides will work, but just pick one and justify your choice. Alright, I'm going to pick the second result from CHOPCHOP, and it's the third one in CRISPR. It has the least number of total off-target hits, and specificity is at the top end. According to CHOPCHOP, its GC content seems balanced, and the predicted efficiency is also on the high end. Clicking on this sequence will bring you to the Focus tab. This visualizes the result. The guide sequence is right here, and it goes from right to left in this case, meaning that it is on the reverse strand. CHOPCHOP also has this list of primers, and as you can tell, they are asymmetrically flanking the guide region. These primers are helpful for surveyor assay used to empirically test the efficiency of the guide. If you scroll down, you have predicted off-target hits with a nice visual of mismatches. Now, if this is the PAM sequence, then we expect the cut will be made 3 nucleotides upstream between T and C. This information is highlighted in the bottom section, where a gap is placed to suggest the cut location. The cut site is highlighted in this visual as well. So we have a guide, and now you want to use it. One way to approach this is through CRISPR by clicking on the cloning link under the result. It has a lot of information. There's an expression of guide RNA using the T7 expression from a plasmid, and many other things if you scroll on this page. The U6 expression from a plasmid is the most common, and I will use pX330 as the example. The page will update with the primers you need to buy in order to clone your guide sequence into the plasmid. You will notice that it adds a G to the Watson strand in case there are no Gs in the front. This will be the G20 guide, meaning that 20 nucleotides match the target DNA. Alternatively, you have a choice of removing one nucleotide from the guide sequence and adding a G, and that's a G19 guide, meaning that only 19 of 20 nucleotides in the guide match the target DNA. There is a note up here that you can read about why a G nucleotide is preferred at the start. I will go with the G20 guide and use this set of primers to clone the guide sequence. At the bottom, there is more information on it. It says that pX330 needs to be digested with BBS1, which is a type 2S enzyme, and there's also a link to the protocol you can follow. So, you buy these 2 sequences as primers from your favorite company. And here's what conceptually happens. The guide primers are annealed and ligated into the pX330 plasmid that is digested with BBS1 with compatible overhangs. The final plasmid looks like this. Here's the gRNA scaffold where the guide RNA is ligated. This entire segment forms the sgRNA. The stretch of T's at the end is the transcription terminator signal for RNA polymerase III, because the U6 promoter is RNA polymerase III dependent. Half of this plasmid, as you may have already noticed, is taken up by the Cas9 template, which of course makes the Cas9 protein. This plasmid can be transfected into the cells, resulting in sgRNA and Cas9 protein. Together, they form the active Cas9 endonuclease, which then hunts for target DNA sequence to make double-stranded DNA cuts. If you want more details on Cas9 targeting, check out my previous video, which is also linked below. Guide RNA design is a small part of the CRISPR experiment workflow. I'll go into some technical details on the next steps in the upcoming video, and I will see you there.